Hello and welcome to the show How Does a Molecule Become a Medicine? Brought to you by the Pint of Science team, Nottingham. I'm Sebastian, or Seb, your host, and I'm a PhD student at the University of Nottingham, studying small proteins called cytokines that coordinate our immune system in its fight against pathogens. And speaking about pathogens, I'm sure most of you have had enough from this one little bug that has kept us on our toes for more than a year now. But thankfully, with unprecedented speed, multiple vaccines have been developed that, touch wood, will soon enable us to return to normal lives as before the pandemic. But how exactly does one develop such a drug or vaccine, and what can we do to improve the process and keep up the pace even for other drugs as well? We've invited two fantastic speakers, Dan Bakowski and Peter Scholes from Nottingham-based pharmaceutical companies Signature Discovery and Quotient Sciences, who will take you on the journey a molecule has to take before becoming a fully authorized medicine. Dan will start with his talk on modern early drug discovery and preclinical testing using the antiviral lenacapavir as example. And after this, Peter will take over to tell us all about the clinical trials and how regulators actually decide if the medicine is fit for treatment of patients. And while we're very sad not to bring these talks to you in our usual collection of pubs and bars, Pint of Science chapters from all across the UK have made up for this and created a number of exciting online shows that will be streamed live here on YouTube throughout the 4 days of the festival. So if that sounds interesting, make sure to have a peek into the program at pintofscience.co.uk and also check out the Science Art Gallery in a tab on the website. And finally, this show is made for you, so please get involved by asking loads of questions and commenting using the stream chat. Our speakers will be happy to answer any questions in the Q&A sessions following their talks. And furthermore, please let us know about the most interesting drug-related or drug discovery-related fact you took from today's show by tweeting at Pint of Science using the hashtag Pint21 and hashtag molecule to medicine. And now, without further ado, I'm delighted to hand over to Dr. Daniel Bakowski, Principal Scientist at Signature Discovery, for his talk. Over to you, Dan.
Thank you, Sebastian, for the very kind opportunity to talk about drug discovery and for your very, very kind introduction. So I think to really understand how early drug discovery works, it's probably useful to go back in time first. The roots of medicine, they run deep. And it's easy to be very patronizing about our forebears. I mean, here you can see bloodletting and leeches. It's very easy to sort of mock the state of scientific knowledge then. But it's also important to recognize that even then, there is a history of empiricism, of astute observation. And in fact, it was this astute observation that was the only means by which we used to be able to find new medicines, where essentially it was just a matter of seeing that a novel substance or a novel treatment could exert a beneficial or desired effect, i.e. it made a disease better. And you can hopefully understand the advantage of this, in that it requires absolutely no understanding of biology. It simply requires someone to be able to show that a remedy works. And on the right here, I've got one such astute observation, and this was from 1763, and it's a rather lovely letter to the Royal Society by his name was Edward Stone, and what he showed or he reported was that willow bark, an extract from that, was very good at treating ague or fever. If you actually look at the rationale for why he tried it, it's rather charming and certainly not very scientific, but nevertheless he showed, and this is probably what we could describe as a very, very early clinical trial. And our next stop then from sort of history way back in the past is the late 19th century, 1897 to be exact, Leverkusen, Germany. And by 1897, chemists had identified the active factor in willow bark, and there's a compound called salicylic acid. But salicylic acid, it does reduce fever. It's an antipyretic. It reduces essentially fever symptoms, but it's not a very nice drug. It's extremely bitter and it's very, very corrosive. And so the chemists at Bayer, and it's important to understand this is not only the birth of modern drug discovery, it was the birth of the modern organic chemical industry. What these chemists did is they deliberately synthesized some derivatives of salicylic acid, and they thought, oh, can we improve on its properties? Can we find a better drug? And we might very generously call this a library today. And the chemists, they synthesized a grand total of 5 derivatives. So it's not very ambitious in terms of modern chemistry. And what they found was that one of these, and that's the top derivative, the acetyl derivative of salicylic acid, acetyl salicylate, turned out to be far, far better than the parent molecule. In fact, it was so good, they decided that within 2 years, it was being sold to patients. And it's the world's first blockbuster drug. And even in 2021, 1 billion people a year still take this drug. And what I want to say is that the reason I've introduced this is that it's an interesting sort of process. We have this concept of a lead. Compounds start with, we do a bit of chemistry around it, we generate a library, and then we look for the best of these. Can we improve on it? Screening. And this really is the process of modern drug discovery. This is late 19th century, remember. So, excuse me. But a lot has happened in the 120 years since. And ultimately, it's a question of scientific advances. There have been huge, huge advances in both chemistry and biology. So, if you will recall, the biochemists, they generated a library of 5 compounds. Now, a big pharma company would have a drug discovery library of perhaps 1 million unique compounds. I mean, it's an unimaginable scale. And also, there have been significant advances in biology. In fact, science as a whole has moved at a pace and in directions the early chemists, early pharmacists would probably have unimagined. They simply could not have imagined the pace and the scale of scientific advances. But what this improvement, this understanding in biology really offers is it gives the option of target-based drug discovery. So instead of just throwing chemicals at a disease and looking for a phenotypic improvement, we can actually identify a specific target and decide, okay, we're going to drug this particular protein, this particular enzyme. And it's all based on a scientific rationale by understanding how the disease works. If we can understand physiology, if we can understand pathophysiology, we can understand maybe there's a good way to develop a modern drug. But the modern drug discovery is very, very different from the late 19th century too, in other ways. So first and foremost, it's incredibly slow and it's extremely expensive. Drugs will commonly take longer than a decade to take to the clinic, and easily over $1 billion. And worse, attrition is very, very heavy in this industry. Most projects will fail. And this attrition, because of the huge cost and time investment required, it discourages innovation and inflates drug prices. We're all familiar with stories of big pharma and drugs. So this has led to the concept of, well, okay, we have to accept there is attrition from various reasons. So what if we can fail fast, fail smart, and fail cheap? That way, we can recognize that there is attrition, but we can mitigate it. And that's understanding the process, having a good drug development process. We can actually reduce the impact of this and hopefully get drugs to clinic faster and more cheaply. So what does the modern process of drug discovery look like? So let's just recall it takes over a decade and billions of dollars to get drugs to market. So I've simplified it somewhat here. It's just the 4 initial events. And it's a process with many moving parts. So first and foremost, for target-based drug discovery, we need to have a target. We need to have confidence that interfering with this target, inhibiting it or stimulating it, will give us a beneficial effect in a disease. And I hope everyone here will appreciate the importance of basic research for this. We need to elucidate the biological pathways, which let us understand the molecular mechanisms of disease. And by understanding those, we can then identify vulnerabilities in those diseases, places were soft points where perhaps we can intervene pharmacologically. This is all hypothesis driven. I mean, science drives every aspect of modern drug discovery. We need confidence that modulating this target is going to work. But having a target is just part of the puzzle. The next thing we need to do is to have an assay through which we can interrogate this target to see whether drugs work, whether or not they inhibit or they stimulate, depending on the target identification and validation. We might want to inhibit some, we might want to stimulate others. And primary assays, they come in many, many flavors. They can be biochemical, let's say purified protein, the target itself. They can be cell-based, and they have many, many different readouts. But what's the important thing they all share is that they are very, very robust, they're reliable, they're efficient, they're miniaturizable. Because what we want to be able to do, ideally, is throw hundreds of thousands or millions of compounds against this target to see what hits, what works. And this is something we call a high throughput screen. So following a primary assay, we'll then have secondary assays. And these come in, again, many, many different flavors. We'll have a selection of orthogonal assays. So these just give us confidence that what the primary assay is telling us is true. If we've identified a novel chemical compound that looks like it's hitting our target, we need to confirm, is that genuine? Or is it some peculiarity of the assay itself? Is it a false positive? So secondary assay will give us confidence there. They will also show us whether any compounds are toxic to cells. Obviously, a toxic compound, it's probably not going to make it into the clinic. It's not going to be a very nice drug. And finally, selectivity assays. And that's because drugs are very, very promiscuous things indeed. In fact, it's vanishingly unlikely that a given drug will only bind to and modulate the target we're interested in. So target-based drug discovery, for each target we're looking at, there are tens of thousands of off-targets where we might not want to drug. For example, some of the proteins, the ion channels involved in regulating the heartbeat. We clearly don't want to target those with an immunomodulator, for example. And finally, the final stage is something we just call ADME DMPK, which is fantastically confusing to anyone new to the field. But ultimately, all it means is drug fate. So for a drug to work, we have to be able to deliver it to the target. We have to get it on board the patient. We also want to make sure that it stays in the patient for as long as we need to have it exerting an effect. Many drugs that are fantastic in early in vitro assays, in the primary assay, in the secondary assay, they would never ever make it into the clinic because some of them will simply not be absorbed by a patient, or others will have a very, very short half-life, which means they're cleared from the body, they leave the body very, very quickly. They're either excreted or they're metabolized. So having an understanding of this is crucial to modern drug discovery. And finally, the most important thing about this cascade, as we like to call it, is that it's iterative. So what we can do is we can throw 1 million random compounds at a target, and we'll get a few hits, a few thousand compounds that are quite interesting. But what we can do is we put those through this cascade, and we tell chemists, okay, these compounds are interesting. They're not good drugs. They're not very potent. They're not very selective. But can you do anything with them? And the chemists will then tweak, they'll modify them, just like Bayer did for salicylic acid. And then we put those back through these assays, and we say, is it any better? Just like Bayer did. And ultimately, with enough iterations here, hopefully the goal is to get a much better compound, a much more drug-like molecule. And with each iteration, each generation that we operate this, we improve our knowledge around the drug, this concept of structure-activity relationships, to understand how the chemistry works, how it binds to our target. And we've got access to all sorts of magnificent, fantastic scientific techniques. We can actually visualize drugs binding to targets with techniques like cryo-electron microscopy, X-ray, crystallography. Really, really fantastic. These are things that biochemists would never even have imagined was possible. So I want to give you a case study now about modern drug discovery. And I've selected this compound here, lenacapavir. I'm pretty confident that not many of you will have heard of this compound. And so why? Why pick this compound that no one's ever heard of? And there are 3 reasons. The first is that it's an antiviral. And what 2020 and 2021 is still showing us is that everyone is trying to become a virologist almost. So really, it's an interesting target. It's also a chance to rehabilitate small molecules, which is what drug discovery is largely about, as antivirals. And for COVID-19, it's been a very, very complex, murky past. Everyone here will have heard of hydroxychloroquine, ivermectin, zinc being sold as cures, panaceas to COVID-19. And the truth is they simply aren't. They simply don't work very well. And a proper discovery cascade would show this, and it has shown this. And yet still we see these compounds having traction when they really shouldn't. Lenacapavir was also voted Small Molecule of the Year in 2020. So it's an exciting, interesting molecule for many reasons. And finally, there are quite a few twists in the discovery tale, which is quite nice. So let's step back to this process of drug discovery. So target ID and validation. What is lenacapavir meant to be doing? So what I've got here is a very, very simplified life cycle of the HIV virus. So lenacapavir, it's an antiviral used for the treatment of AIDS. So it's obviously a very, very different viral disease to COVID-19, but one which is of probably very similar sort of burden to society. It's a catastrophic chronic disease, heavily stigmatized. And this is the process, the life cycle. And you have to understand that we've got huge knowledge about how this virus works. And each of these steps, this is the virus binding to a target cell, being imported, releasing its genetic material and enzymes, and then going through the process of replication, budding, and then generating many, many more viruses to infect many other cells. By understanding the steps here from basic science, each of these is a vulnerability that we can potentially target with drug discovery. And the target that lenacapavir has picked is quite an interesting one. It's this thing here, the capsid. And the capsid is essentially the packaging for the HIV virus. It's a really beautiful structure. It's a fullerene cone, and it's a polymer basically of a capsid protein here. And it forms a little protein coat that contains DNA and enzymes. And the rationale is, well, it's quite obvious. If we can somehow prevent this coat forming, we can immediately interfere with several steps in the HIV life cycle. So it should be an antiviral. And this is a program that was run by Gilead, and all of this data is in the public domain. It's well worth reading out. It's a really fascinating story of drug discovery. And the assays that they used to support this was an in vitro capsid maturation. So they simply took purified protein and thought, oh, can we stop it forming these capsids? And the first twist is, no, they couldn't. They couldn't stop it forming with inhibitors. They just couldn't generate compounds that are active enough. But does this mean the program failed? And the answer is no, because there's a very, very interesting finding, and that was that they could generate accelerators of capsid maturation. They could actually give these proteins to form this coat a helping hand. They could accelerate it. And it turns out that this is also a very, very effective antiviral mechanism. In fact, much more effective than you might intuitively think. By giving the virus a helping hand, the capsid forms so quickly, it does not form properly. And so it's not a competent package of the RNA, of the genetic material and the enzymes of the virus. So the virus cannot replicate. It cannot do its normal life cycle as well. So this is the first lesson that lenacapavir provides. We get unintuitive mechanisms of action. But this drug discovery strategy could pick that up. Again, it's this understanding that it's informed by basic science, understanding what we're doing and how we're targeting. So this slide is basically, I want to tell you another little sort of trick that lenacapavir shows. And this is lenacapavir. I mean, look at the complexity. It doesn't take a medicinal chemist here to say, whoa, this is a lot more complex than aspirin. I mean, this is a really, really complicated molecule. And it turns out that over decades of drug discovery, chemists have sort of built up a selection of rules or more guidelines as to what constitutes a good drug. If you will recall, if you want to make a good drug, we need to get it into a patient. We need to be able to deliver it. And chemists have come up with a list of rules, they like to call them, or guidelines, molecular descriptors of the physical properties of a drug that make it an effective oral drug, something you can take every day and it's going to be absorbed. That's the big, big challenge for many, many drugs. And you can see that lenacapavir, it violates most of these rules. In fact, it violates them really quite severely. I mean, it's a huge, huge molecule. So what's the twist? What does lenacapavir tell us here? Does it tell us that these rules don't work? No, the rules are fine. Lenacapavir is a truly terrible oral drug. But there's the qualifier, oral. Although it doesn't work as an oral drug, you can inject lenacapavir. And in fact, what is remarkable is that you only need to inject it once every 6 months. 6 months. This is a drug, it's fire and forget. And I think that's the most remarkable thing about this drug. It's an appreciation of, by the biologists, the chemists, of the disease that the drug was being used to treat, the indication. HIV/AIDS. Remember, this is a chronic, lifelong condition. It's heavily stigmatized. And for a normal oral drug, if someone forgets to take a dose one day, there's a chance that you can get resistance evolving in the virus. There's a chance of viral transmission. But here, you simply have one injection every 6 months, and you have this sustained antiviral activity. And so, yes, so I think that's a key thing to remember here. It's this understanding that drug design is not about just generating a molecule that can potently stimulate or inhibit your target. It's about understanding, well, how will this drug be used in the clinic? So yes, lenacapavir, deservedly small molecule of the year. I've just put side by side aspirin 1897 and a century and a bit later. This is what medicinal chemists are capable of. And it's just completing a clinical trial at the moment, and it's looking extremely promising. It's first in class, really exotic chemistry, incredibly potent, novel mechanism of action, a reminder that drug discovery is about understanding where you can bend rules, where you can break them, and yet still get a fantastic drug out at the end. And above all, it's an understanding that modern preclinical and clinical drug discovery is an appreciation of what you are trying to treat. So I shall leave that there. And if you do have any questions, I'd be happy to try to answer them. Thank you.
Thanks. That was a great and very interesting talk. Yeah, I didn't realize that that drugs can break so many rules and still make it to the clinic. Yeah, very interesting. So as Dan already said, if you have any questions, please type them in the chat, and I'm sure Dan is happy to answer them. But I've actually got a question myself, not about lenacapavir, actually, though. So you've mentioned in the early part of your talk that the failure rate is very big. So the common thing to do for pharmaceutical companies is now to fail easy and fail cheap. But is someone looking at bringing this failure rate down, or is that basically left?
There are many efforts in trying to understand how drugs fail. And it's a very interesting, if you look at the reasons for why drugs have failed, historically, it was largely toxicity. And often this would be idiosyncratic toxicity, that only once you get it into the clinic, you start realizing that your drug, unfortunately, may kill people more than it can help them. And there are, of course, many stories of drugs that have failed in this way. Interestingly, for modern drugs, they tend to fail because they don't work, which is very, very interesting. And this is why the concept of target identification and validation is so important. The idea is we have to do this properly to understand that, is our target really involved in a disease? And also, are we looking at it? Are the models we have of disease, are they adequate? And the simple answer is that in many cases, they're not. They're not predictive. But there is a change. I mean, it's nice that drugs are failing for different reasons, but clearly there's still a lot of work to do.
Yeah, I hope, because it will obviously also bring the cost down, wouldn't it? Yeah, so, yeah.
An attrition rate of high 90%, programs that cost tens of millions of dollars before you reach that critical failure point. It's a very uncomfortable position to be in.
Yeah. Okay. Thanks for that. So I can see we've got quite a few questions in the chat. We'll answer a few of them. I'm not sure if we can answer all of them. Okay, so let's start with Gemma, who asks, do you think that AI can help with target identification?
Yeah, that's a very, very interesting question. And the answer is yes. Simply by as we understand the biology more, we can do sort of ontology. We can understand which targets appear to be associated with certain diseases. We can do a genome-wide association studies to identify this. And of course, this is big data. It's absolutely fantastic for AI. But I think in the short term, the contribution of AI is more likely to be in the medicinal chemistry side, to make these esoteric leaps in chemistry to say, well, what about trying this different molecule? It might bind, it might be an improvement of what you already have. Is that better than the medicinal chemist? There have been some lovely papers showing that AI can actually be just as good as a medicinal chemist. I don't know, to all my medicinal chemist friends, I want to say that yes, we still need definitely human eyes to look at these things.
Yeah, probably they will work next to each other at some point, hopefully. Okay, let's see what other questions we have. So Sarah asks, I was wondering, in what ways do you think it will be possible in the future to shorten the time it takes for molecules from when it's discovered until it reaches the clinic?
Yeah, that's another very good question. We know it's possible. And of course, the example of COVID vaccines. We can get medicines into the clinic incredibly quickly. I think it's once we have confidence in the safety studies that we can get drugs quickly through. The long-term effects, it's clearly there will always be questions about that. And lots of the problems we've had historically have been these longer-term emergent problems with drugs, these idiosyncratic effects that early discovery, early clinical studies simply aren't powered to detect. But it is possible, certainly. Unfortunately, as we get more and more scientific techniques, there's more and more science we can do in this early drug discovery stage that people will expect to be able to do before we can advance to the preclinical stage. So in a way, we're kind of lengthening the drug discovery process. There's a danger of that too.
Okay, great. And I think we've got time for one last question. Paramita asks, how much can organoids help in drug discovery? And what are the caveats that organoids bring? And are they actually used already much in the discovery process?
Yeah, another nice question. So organoids they're an attempt in an in vitro system to try to recapitulate a target organ. So many of the criticisms that are very, very fairly thrown at drug discovery projects are the assays we use. They're not predictive. They're not close enough to a living person. They're remote from the true, genuine physiology. And an organoid is an attempt to try to correct that gulf of physiology. So organoids, an organoid is like a bit of a gut. You can use its stem cells to form gut-like organoids, where hopefully you've got a much more native-like tissue. And if it's more native-like, then you hopefully expect it to be more predictive for drug discovery. But this comes with a cost. And the cost here is one of opportunity and the time it takes to run these. So they're not very good for doing the kind of large-scale, high-throughput screening. But they're absolutely, probably really, really very, very powerful for later stage discovery. When you've identified your hit, your lead, you know you've got an interesting chemical series and you really want to understand it better, then they're very nice. Yeah. Good question.
Okay, thanks. And unfortunately, we won't have time for any more questions. I hope we can get these questions answered in another way. I think we'll probably figure something out. But for now, thank you, Dan, for your exciting talk and answering all these questions in such great detail. It was really nice to have you on.
It's a pleasure. Thank you for inviting me. It's really enjoyable. Thanks.
Thanks. Okay, and now, before we continue with our next speaker, we will explore some of the rather unconventional ways how drugs have been discovered. I think Dan has mentioned it, and many of you will know that some of the most famous pharmaceuticals actually didn't undergo the classical drug discovery process, but were discovered purely by accident or through the occurrence of side effects during treatment of another disease. And to test your knowledge on this, we've created a small quiz which I would like to invite you all to participate in. So if you know the answer, please type it in the chat and let us know how well you did at the end. Okay, so let's dive right into the first question. I think that's a fairly easy one that many of you will know. So question 1 is, who discovered penicillin? Once again, type the answer in the chat if you know it. And I'm quite sure many of you know the story behind this as well. So that person that we were looking for was a microbiologist working on bacteria, and at some point when they checked their petri dishes with where bacteria usually grow on, they discovered a fungal contamination, and that had a bacteria-less region around it. And it turned out that the fungus released penicillin to kill these bacteria. So let's have a look in the chat. I can see a lot of questions already. Most of you, a lot, said Alexander Fleming, and that is, of course, correct. Yeah, he was the discoverer of the first antibiotic. Okay. So question number 2, which is a bit harder. So the blood thinner warfarin was derived from which plant? Was it A, fennel, B, sweet clover, or C, milk thistle? I think that's a bit harder. And the reason why you can see cows on this on the screen right now is the story behind this, because it was farmers in the US and Canada that complained about their cattle bleeding to death after just undergoing minor procedures such as dehorning, and it turned out all of these cows had eaten from the moldy plant in question, which contained the active compound decumarol, and this was then further investigated and eventually led to the development of this blood thinner that saved countless lives. Okay, let's see in the chat, a lot of people responding. I can see a lot of B's. Someone said C as well. Okay, let's see who was right. It was indeed B, sweet clover. Yeah, who would have thought that cattle were the discoverers of a modern day drug. Okay, so for our last question, we have a quite funny one, I would say. For which disease was Viagra originally intended for? So was it A, angina, was it B, depression, or was it C, Alzheimer's disease? And so I personally found that the story behind this quite funny. So the Viagra was originally actually synthesized in the UK in one of Pfizer's labs, and when it underwent clinical trials for the original disease it should treat, a very observant nurse reported that a lot of men were laying on their stomachs when coming into the clinic for checks, for obvious reasons, I guess. And then Pfizer soon realized that this is an excellent way to treat previously unmet need, to treat erectile dysfunction. And so it became the first oral drug for this. Okay, let's have another look in the chat. I think the chat is saying A, angina. I think there's a couple of people that think otherwise, but let's see what the right answer is. Yeah, so you were right. It's indeed an angina. Great. I think everyone did very well in this quiz. Let us know how well you did. And I'm sure you appreciate that sometimes luck can beat the most elaborate scientific method. to discover drugs. But nevertheless, even the drugs that were discovered by accident have to show that they're safe and effective before making it to the market. And you've probably guessed it, that's exactly the topic for our next talk. And I'm really excited to welcome Dr. Peter Scholes, CSO at Quotient Sciences, who will tell us all about clinical trials and the late stages of drug development. So over to you, Peter.
Thank you, Seb. Hi, everyone. Thank you for the opportunity to present and virtually meet with everybody. It's a shame we're not all down the pub, of course, but hopefully next year. It will be fairly whistle-stop tour through clinical development. Thinking about the best place to start, and as always, it's a case of start with the end in mind is a good maxim, a good saying. What is the purpose of clinical research? What's the final outcome from those labours over many years? And in some respects, it's shown on the slide here, albeit just for one of hundreds of medicinal products, of course, but a local one here in Nottingham that was invented, discovered by Boots, ibuprofen, brand name of Nurofen, of course. But the two screenshots are really the outcomes of many years of research in terms of information that's given to patients and information that's given to prescribers and physicians. So first of all, we have the patient information leaflet. That's a little bit of paper in our packs of medicines we get from the pharmacy or the doctors, which I'm sure we read rigorously. But in essence, it explains to the patient what the drug's for, how it should be taken, how it should be stored, what potential side effects are. And the summary of product characteristics, the SMPC, is a more detailed, I guess, more scientific version of that for the medical doctor, which goes into more detail on the therapy areas, the dosage regimens, how the drug should be given in particular populations, for example, but really explaining how hopefully that medicine can be prescribed to that patient to cure the disease or relieve the symptoms. And really everything we do is aimed at, yes, it's gated by regulators, but it's aimed at providing the information to allow drugs to be prescribed safely to treat or indeed prevent disease. As Dan's already said, it's a long journey, typically 10 to 12 years, and Daniel did a great presentation on the left-hand side covering discovery and preclinical into clinical trials. I'll cover that bit in the next few slides, of course, before the end of the runway, reviewed by regulators prior to market approval, and when a new drug becomes commercially available for patients. I think the thing to note here is, and there's some numbers which start to illustrate this attrition, this high failure rate. I think Dan mentioned 98%. But you can see here, you start with thousands of compounds in the laboratory just to get one product on the pharmacy shelf. And time often equates to money in life, of course. And just some numbers to reflect that. This was quite a key publication from 2016, where some researchers used a really elaborate pharmacoeconomic model to understand the cost of drug development. And this is both the cost to get a drug to market, but also it includes the cost of all the failures on the way, all those that don't make it to the destination. And the estimates then in 2016 was over $2.5 billion to get one product to market, which again, without necessarily seeking to justify, starts to indicate how the cost has to be recuperated somehow on that journey to fund the next generation of innovative medicines. Other research papers more recently have indicated the cost could be higher. Bottom line, it's an expensive process. The good news is pharma and biotech industries are not dissuaded by that. And the left-hand plot here shows actually there's more potential new drugs being researched now than ever before. Indeed, there's over 18,000 in development as we speak across the globe at various stages to bring new products through to market for unmet clinical need. The right-hand side is the sobering view of new products that actually get over the line. And looking back over the 10 years, this is in terms of new drugs approved in the United States, the average is less than 50 per annum. Perhaps in the last couple of years, there's been a slight uptick, but again, a further emphasis of the need to get more efficient for we as an industry to move more products from the left to the right and get them into the hands of patients to treat unmet clinical need. So what's required ultimately to get a new drug to market? I'll mention a few times about the independent regulatory oversight. So what do regulators look for before they will sanction a product as being suitable for being prescribed? And there's three main pillars of that: the safety, efficacy, and the quality. The safety, Dan's started to describe the fundamentals about the drug interacting with the target and minimizing off-target interactions or side effects. The efficacy are really the fruits of the clinical research, which I will describe shortly. Does the drug work clinically? Do the benefits overcome the risks for those patients? Quality perhaps doesn't get as much airtime, but in essence, that's around the product, the tablet, the inhaler, the injection that gets administered to us. How can that be made to a high quality and a reproducible standard? And the beauty of live events, that's the phone, I do apologise. And indeed, how can the product on the pharmacy shelf be reflective of that which was used in clinical research, which proved the product worked in the first place? How do we tie that together? But first, clinical development. A lot of detail on this slide, which I won't necessarily read out. But up until the point of the regulatory review, there's three really clear phases: phase 1, phase 2, phase 3. And we can see here the time base to those: 12 months for phase 1 research, 2 years for phase 3, typically 3 years for phase 2, typically 3 years for phase 3. But also, as we go through those stages, there's a shift from healthy volunteers in phase 1 research, typically in small numbers, less than 100. Phase 2, we get into hundreds of patients, and then phase 3 in thousands of patients. And the purpose of the research is to answer some fundamental questions in order to get that product to market. There's a recurring theme of safety. The intensity and the scrutiny is relentless. Is the drug safe at every stage of clinical development? In phase 1, though, the focus, in addition, is really understanding what the body does to the drug. These are volunteers, they're not patients, but can we understand how the body processes the drug, how the body eliminates the drug? In phase 2, when we're in patients, we can start to study how does the drug work? Is the pharmacology working? Is what we hoped what would happen based on computer models or animal models? Do we start to see those interactions clinically in vivo? And how much drug should be given? What's the dose? For how often? For how long? To start to answer some of those fundamental questions. And then phase 3 are the larger trials, multinational, multi-center, thousands of patients, often comparing the drug to a placebo or the current best practice in care. You're really to prove once and for all the therapeutic potential for that new molecular entity. But following the regulatory approval and hopefully the commercialization of those products, research can indeed continue. Safety is always still monitored, and other indications perhaps can be pursued for that medicine. The last row, I think, also picks out just one final time: attrition. And this is a real, hopefully, simple representation. If, for example, 100 molecules make it as far as clinical research, so go through the work Dan described, then at the end of phase 1, on average, 64 will still be alive. At the end of phase 2, 20 will still be in development. And then 12 will go to the regulators and 10 may get approved. So basically, there's a 10% chance of success, even if it gets as far as phase 1 clinical research. And by the end of phase 2, 8 out of 10 drugs will have failed. So that's a real focus for us, I think, a real opportunity. One of the earlier great questions was, how can we improve drug development, reduce attrition? So some questions perhaps to help answer that from a clinical perspective. How can we start human trials quickly and show the drug is safe, perhaps faster? How can we get that new drug into patients sooner to see if it's going to work? Again, in terms of time reduction, cost reduction. And don't forget the product. We talk about drug as a generic term, but from a pharmaceutics perspective, we would view that as the white powder. The product is actually a formulated composition of that drug, which is delivered to the patient, and often contains other ingredients to achieve the desired therapeutic outcome. Most drugs are delivered orally. Obviously, the example Dan showed ended up being an injection, but most drugs are delivered orally. And if we just think about human physiology and human evolution, and there's a little schematic there to show esophagus into the stomach, into the small intestine, the colon, we really need the drug, unless it's for local disease, to be absorbed into the bloodstream for the drug to work. And if you think about it, the reason we exist today as human beings is that evolution has helped our gastrointestinal system be a tremendous barrier to foreign chemicals, to stop poisons being absorbed into our bloodstream, which is great on one hand, but not great for new medicines. So pharmaceutically, we have to get that, say, the tablet to disintegrate in the stomach, the drug to dissolve, and then the drug to be absorbed across biological membranes into the blood. And just showing one graph, I can't help myself, is a typical, what we call a pharmacokinetic profile. This is the blood concentration in the plasma against the time course over, say, a 24-hour period after taking that tablet. And this would be ideal. Rapid absorption, not too much drug, so the peak plasma doesn't cause side effects. There's then a decay as the body starts to metabolize and eliminate the drug, but it would stay above a certain concentration threshold over 24 hours, suitable for a once-a-day therapy. But life's not like that, of course. Most drugs are poorly soluble. Some drugs are poorly permeable. Some drugs are broken down too quickly. So a tablet is not just a tablet. Often a tablet contains these other excipients, ingredients, to try and overcome some of those challenges that we have to live with in terms of drug chemistry. So I was trying to think of a metaphor for the talk today about, again, ways we can go about addressing this. And hopefully it's apt and meaningful on a day like today where rules relax and social distancing eases a little bit. Thinking about going to the local cafe or restaurant and to order a meal, and we expect things to come quickly, don't we? The ingredients are there, the product's baked there, and we eat it there. And if we don't like it, we'll send it back to the kitchen. But the pharmaceutical industry isn't integrated like that. We have the drug substance, the white powder, there's the product to develop and make, and then we ingest, we test it clinically. But they're very discrete activities, often in, or typically in discrete locations. And what that can mean is a very convoluted supply chain and a lot of time to get a drug turned into a product to dosed in people, often 12, 18 months actually. And the result of that can in effect be a game of snakes and ladders, that if the drug and the product doesn't elicit the response we need, we have to go back to the start and develop another one. So it's a very, very inefficient process because of these silos. So again, there's a lot of industry interest about breaking down those silos. And here in Nottingham, we've been a pathfinder in some of that, and really integrating the manufacturing of pharmaceutical products to a high standard, and then the clinical testing of that, and really doing that in a matter of days rather than a matter of months. And at Quotient, where I work, we've run over 400 studies over the last decade with that very model, making and dosing quickly and safely. And that's driven a lot of documented time and cost savings. But scientifically, of course, even better than that, we use human data to make decisions, human clinical data, those pharmacokinetic data, or biomarker data if we're looking for some assessment of pharmacology, rather than in silico computer models or animal models, which run the risks of not being predictive for the human response. So then we can start to re-engineer early clinical development, for example. So the left-hand sketch shows a typical first-in-human study in healthy volunteers, what's called a single ascending dose, where the dose of a new drug is escalated carefully, slowly, and with high medical observations. And as well as doing that, what we can now do is introduce the ability to look at different formulations of that drug, if needed. You look at different technologies, if, say, solubility is a problem, or permeability is a problem, or the metabolism or half-life is a problem. So in phase 1, if you remember, yes, we answer the question, is the drug safe? Is it tolerated by humans? yes, we understand the pharmacokinetics, but we also gain great insights early into the product, so we can make and supply product with confidence into those initial phase 2 studies. So we can, in a better way, look for signals of efficacy in patients. And all this, I hope, will start to reduce that attrition rate of 80% of drugs failing by the end of phase 2. So a quick case study. This is work we did at Quotient in Nottingham for a San Diego biotech called MEI Pharma. They had a new molecule heading out of discovery into clinical development. But there were solubility concerns. There was uncertainty as to what formulation they should make, which, remember, could take many months to dose in humans to make sure the drug would be absorbed and be bioavailable. So by integrating activities, yes, we did some computer-based modeling and some laboratory formulation studies, but we identified 3 potential formulations to dose in this first study. Without going into too much detail, one where we reduce the particle size of the drug to make it dissolve faster, one where we include fatty lipidic excipients, which can improve the solubility, and one where we turn the drug from a crystalline form, which often needs a lot of energy to dissolve to be absorbed, into an amorphous form, which is a better energetic state for the compound. And then in the first in-human study, we had the chance to look at these different formulations as well as ascend the dose. And the good news is we found a relatively simple formulation which gave adequate bioavailability, so adequate levels of the drug in the blood, which were verified by a biomarker, which the MEI hoped would be predictive of efficacy in patients. And that allowed us then in 12 months to get phase 2 testing underway with a drug product which maximised the potential for success. And this drug is in phase 2 trials today, and fingers crossed, enjoying some successful results in some rare blood cancers. So hopefully in a few years' time, this will make it to market. Last minute or so, just mention about regulatory oversight of the whole process, not just phase 1 trials or phase 2 trials, but the final regulatory review prior to marketing authorization. In the UK, our regulatory body is the MHRA, the Medicines and Healthcare Research Authority, and each clinical trial needs a separate clinical trial application. And there's a sister process in the US overseen by the Food and Drug Administration. So sponsors of studies have to submit a lot of information for every clinical study about the drug substance, the drug product, the previous clinical and non-clinical history, and the clinical protocol they wish to execute and perform to answer the next question during development. But each study doesn't just need regulatory review and approval, it also needs independent ethics. And again, in the UK, there's independent ethics committees which review similar information, but look through the lens of the volunteer. Look at what information the volunteer will be given, make sure the volunteer, whether they be a true volunteer or a patient participating in a study, make sure their welfare is protected and their safety is assured as well. So my last slide, hopefully what we've gone through, both Dan and myself have indicated that your medicines development, it's a long journey, it takes a long time, costs a lot of money, and there's high failure. But as a complete industry, we are looking ways to improve the efficiency of drug development while staying within a really robust system of checks and balances. And really integrated research, I think, is a way of breaking down some of those barriers to reduce time and cost and maximise potential for success. And of course, and today being a case in point in that, and the last 12 months, certainly putting us all under magnifying glass, the importance of new medicines has never been more, had more attention than it does have today. And with that, hand back to yourself, Sad, please, for any questions in the last 5 minutes.
Great, thank you. That was very interesting, and I actually didn't realize that Nottingham, or specifically Quotient, is on the forefront of clinical testing and new developments in that area as well. So I've seen we've got a lot of questions that have come in. I think we'll go straight on to these. We don't have much time. but we'll try to answer as many as possible. So the first question, would you expect aspirin to be approved today?
That's a good question. I hope it would, yes. Again, it would, as with any new medicine, the same is true past, present, and future. It's those basic tenets. Is the drug safe? Is it efficacious? Do the benefits outweigh the risks? And also, is it a cost-effective treatment compared to other standards of care? So it's those same litmus tests, if you like, those same basic questions which the clinical development plan would have to be geared to generate the evidence for. So I'm sure it would be the case for aspirin to be approved today. It still plays a key role in the treatment of some illnesses and some aches and pains, as we know.
Yeah, I think especially on a day where there's quite a few of relaxation in the UK regarding indoor pub, visits and stuff, that would help quite a bit. Okay, so next question. When an existing drug is found safe for one illness and is then found to be useful for another illness, does it have to be tested all over again?
Potentially, or just parts of it have to be. Again, a great question. So if it's the same product, the same tablet, the capsule, no, that's safe. That's a high quality product. There'll be say 2-3 year shelf life. So the quality is already boxed off. If it's the same route of delivery, then you typically the same. No additional safety or toxicological testing would be needed. What would have to be performed is that other leg on the stool, the efficacy assessments. You would have to do trials in that new additional patient population to prove the drug works. And again, if the product changes or it's a different route of administration, then other work in those other areas may be required. So again, it will depend on the level of change that would be required.
Okay, thanks. And I think we don't have time for any more questions. So thanks again to you, Peter, and also to Dan for your lovely talks. And also thank you to everyone behind the scenes who have worked incredibly hard to make this all possible. So there's plenty of more shows coming up in the next 3 days of the festival. So make sure to check out our website for the program in the next few days. And also, please let us know how you liked the talks and what you've taken from them by tweeting Pint of Science using the hashtag Pint21 and hashtag Molecule to Medicine. And I think that's it. So thanks again for tuning in, and have a lovely rest of your evening from everyone in the Pint of Science Nottingham team. And see you next year, hopefully in person again. Bye.